And now we'll go to Paul Francois, who is coming to us from McGill, who is also interested in dimensionality reduction and how to identify latent variables from high-dimensional data. Paul?
Okay, so thanks a lot for the invitation. So I'm going to tell you about some recent methods we have developed in collaboration with Grégoire Altan-Bonnet at the NIH. And since I don't have much time, I just want to acknowledge right now the students who have been working on it, Suraj Asha, Angela Lee, Thomas Rademacher, and Francois Bourassa. So I just start with the kind of, general view of the problem we're trying to study. So in an ideal world, you would have high throughput experimental method to generate this kind of, data that we are familiar with, say UMAP representations or complex interaction networks. So say protein interaction networks. And then, so I'm a physicist, so I'm interested in trying to get a sense of what happens mathematically. And then we will use various methods to try to model what is happening here. So for instance, machine learning methods or just direct modeling of interactions. And the goal here is to do many things. So first, one thing we could do is to try to generate predictions, new predictions from this data, or we might be interested in getting more into things like mechanisms or principles of organization of such system. Now, as a physicist, as a theoretical physicist, one of the big problems is happening here. It's really not clear how to use, how to basically go from this big data to the actual of this scheme with prediction mechanisms. And I'm sure you've already discussed some of this in the previous talks, but I just want to point out some problems that I'm particularly interested in. So one of the fundamental challenge is what I call the connectionist nightmare. Like you have all these things interacting. So for instance, on the top right, this is an actual protein interaction networks for T cells. And you have like all these things talking to one another. When you try to make a mathematical model in the bottom left, you have like tons of equations and it's really difficult to understand what is happening. There is the same problem in machine learning. So if you look at this deep neural network, it's very difficult to understand how they actually work. And one thing I really like to point out that some people don't know much about this. This is this problem of adversarial examples, where in machine learning, classifications are actually quite brittle to noise. So for instance, you have this panda image, you perturb it a little bit, and the classification algorithm is going to classify it as a gibbon. So you really have issues like this where the things are a bit fragile. Another problem, which is really related to the talk we just had, is what is the right way to think about the data. So as a physicist, I like to talk about this heliocentrism versus geocentrism view, where you get the same data, and depending on the way you plot it, you're going to have very, very different behaviors. And it's not, of course there is one that is preferable to the other. Heliocentrism is much simpler to describe than geocentrism, and obviously we like to be in more like heliocentric view when you're looking at actual data. Another problem we have is that, for instance, if you use unsupervised method like UMAP, like here, for instance, on the top right, you have a UMAP 3D representation of a bird. When you go to 2D, you just don't recognize the bird at all. So how do you know which representation to choose is really a challenge per itself. And then, of course, there is a time component. Like if you look at a differentiated cells or a dynamical system in biology, how do you represent something moving in 2D, say, in a low representation? And that's complicated. So this is a movie of a learning algorithm that we've developed with Puzzle. Yes?
We're not seeing your slides advance.
I see. Oh, that's annoying. Do you see it now?
Yes.
Okay. I guess, maybe this one is the most important. I really want to focus on the best way to represent the data. And so when I'm studying complex system, like planetary motions, the way you're going to represent data is going to be important and really to change the way you think about those problems. And so I hope it's still working now. I just want to introduce a general approach we've been using. And so our original approach is to first take your data and then reduce that dimensionality in a supervised way. So we've been developing many different things. So reduction algorithm to take a big networks and reduce them. We also been using evolutionary algorithm to understand biological data and identify the most relevant feature in the data. And then once you reduce the dimensionality of complex dynamical system, we can model the dynamics in this slow space. And then the idea is that once you have reduced the dynamics, you can expand it, go to higher dimension, and reconstruct the biological property. And so I'm going to illustrate this approach on something we've been working on, which is cytokine dynamics. So we can use this approach in developmental system, but also like recently we have been working a lot on cytokine dynamics. And so the idea is that you do have in your immune system, you have T cells that are interacting with one another. And then they produce cytokine, and then the cytokine tell you how the cell, can interact and how they should react to the actual antigen. And then what happens is that you can have, many things that can happen. You can vary antigen concentration. You can vary you can vary T cell numbers, and then you will have, this kind of very different behavior. So this is cytokine called IL-2, and you see like sometimes going up and down, sometimes just going up. You can really very different parameters of this. So there is really an idea that there are many dimensions this problem that you can vary to describe cytokine dynamics. One important parameter is the ligand strength, like how strong the immune response is. And the reason why it's important is because depending on the ligand strength, you might have better response or not. And this is important, for instance, for immunotherapy. And so we have developed a pipeline. So this is with my collaborator, Grégoire Altambonnet, where we try to have a robot to really study in test tube the actual immune response. And so to make a long story short, we just have, this little test tube. We put T cells, we put different antigens, and then we're just studying a bit. We're just monitoring as a function of time everything that happens. So we have many cytokines. We have several markers. We can do many time points. We can just basically somehow sample the full space of a cytokine response of T cell. And so to give you an idea of the data, this is what we have. So each line here is a different cytokine. Each column here is a different peptide, a different antigen. This x-axis is time, and then the different colors here are different T cell numbers. So we can really very reliably and very precisely generate a map of the way the T cells react in the test tube. And so then from there, we have a kind of highly multidimensional data, and then we try to model that. And so this is where, we have to develop new techniques. And so, and this is where we are using things like evolutionary algorithms. So to make a long story short, this movie here illustrates the data that are uploaded in 3D, with 3 cytokines, and you see these different lines are different antigen, and you see these lines are, they are both simple and complicated. They cross. It's a bit like my geocentric thing. But now if we move what we discovered, that we use our evolutionary algorithm, and we discovered that we can find a better way to represent the data. And basically, we discovered that the integral of the logs is the better way to represent the data. And so now if you look at this picture here, the different antigen follow very nice trajectories in this integral of log space. So we basically change the space. And now suddenly we can separate very well the different antigens. The different colors separate very well. And so then from that, you can basically project using like classical machine learning methods. You can project those lines in a simple space. Then you can separate those different antigens. And so you will be able to classify different antigens based on their ligand strengths. And so to make a long story short, I'm going to wrap up soon, but you can use that to do an actual model of the cytokine response. So the first thing that you can reconstruct the full cytokine response from this simple model. Then what you can do is that you can model this trajectory in 2D in ligand space, and you can use a physics tool to actually model this dynamics. And so what is really interesting is that this kind of dynamics that we see in this reduced space, the best way to describe it for us as physicists is to actually use what we call ballistic physics or rocket science, and to parameterize these curves in a way that will allow us to really describe very nicely these trajectories using these parameters. And then, so once we have those parameters, we can actually reduce the model because we like all these parameters are actually correlated to one another, which means that these complicated trajectories of cytokines, you can essentially model them with only one parameter, which is essentially the initial slope. And this initial slope is connected to the way the thing is going to dive and to the initial angle of the trajectory. So we call this parameter immune velocity because it's really like the initial velocity of a rocket that you were going to throw away or to launch. And then once we have this model, so it means that this complicated model was really reduced now to only one parameter. Even though you have a highly multidimensional system with many cytokines, only one parameter can represent it, which is really, really surprising, but that works very well. And then you can test this hypothesis. And so, for instance, what you can do is that you can take other data that we were not using for our training, and you can try to feed those parameters, determine this immune velocity, and see if the model is still working. And what we see is that it works very well. Like, for instance, by plotting the correlation between the initial time and the initial immune velocity, we really see like a very nice structure in the data that tells you that the time, there is this other parameter, launching time, is correlated to the number of cells. You can put new antigens in the system, and the new antigens are basically layered intermediate in between two other antigens, so we can really predict the strength of new antigens. And then you can also go back to mechanisms because now we have a full mathematical model of the response. And then we can go back to the data, such as IL-2, and we can connect this immune velocity to parameters in this curve, such as the cutoff time. An interesting parameter that we discovered is actually we can measure the response, the innate versus adaptive cytokine response that is also predicted by this immune velocity. So we discovered a full new ensembles of connections between the cytokine just based on this dimensional reduction and the fact that this simple immune velocity explains the data. So I'm going to wrap up now, and I apologize for the little problems with the slides, but in a nutshell, what you can do is that you can take this high throughput data of cytokine response, and then you find a way to project them in 2D, and there is a layer here, which is this evolutionary algorithm that we have used to find the best way to project them in 2D. And this is really important. You're not doing it in an unsupervised way. You do it in a supervised way. I do not go too much into the detail, but you have to find a way to really find the best data, the best form of the data to study it. And then we discovered this immune velocity, and we're now using it to test different things. So for instance, we can measure immune velocity for immunotherapy. This is work in progress. So thanks a lot for your attention. And I'm just going to end up, again, thanking my collaborators and showing you trajectories in latent space.